Welcome to the St. Joseph's open web-based learning platform, that is How You Learn. Also welcome to the Department of Biotechnology, St. Joseph College for Women, Autonomous, Visakhapatnam. The Department of Biotechnology, St. Joseph College for Women, Autonomous presents a MOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. It's a free online modular 8-week course for life science background students. The course is being offered for UG level. The course Plant and Animal Biotechnology has 5 modules. The first module is plant tissue culture techniques and second metabolites production. Within the first module, the third chapter is establishment of cultures, Callus cultures and suspension cultures. So this chapter is instructed by Dr. A. Veerabrahmam, HOD and Assistant Professor, Department of Biotechnology, St. Joseph College for Women, Autonomous, Visakhapatnam, Andhra Pradesh, India. So let us see each the detailed course structure and chapter structure within the establishment of cultures, that is Callus and cell suspension cultures. Let us first understand and learn the learning outcomes. The students will be able to understand the basic concept of callus and cell suspension cultures. Here, the callus is a unorganized group of mass of cells will be obtained in vitro conditions. Cell suspension cultures usually obtained through the liquid cultures where the agar is not added, and they will be able to optimize the conditions for callus subculturing. So in callus culture, in order to obtain a variety of products, or in order to study the cellular organization, callus cellular organization, that means the potentiality of the each callus cell and aggregates in plant origin will be uh, identified and optimized, and the students will be able to be able to determine the factors for cell suspension culture. Here the factors are very important, play a very significant role in cell suspension culture, and also evaluate the scalable products of suspension culture on commercial basis. So today, commercial basis, so many firms associated with the plant tissue culture are offering a variety of products, scalable products, and these products are promising the near future in the society, and also able to reproduce the protocols for pilot scale operations. So whatever protocols or innovation or technology concepts learned in this chapter, the the technician or the operator, or most probably the students, can reproduce the protocols in a small scale operations. Let us see the contents in this chapter. First of all, introduction to the callus culture. What is the callus culture? How callus culture can be established? And what are the requirements for the callus culture? Culture material. What type of culture material should be used? And culture material characteristics will be discussed in for the first concept. And the next concept is establishment and maintenance of callus culture. So this is a wonderful area where in case of plant tissue culture, once you establish the callus, you can indefinitely get the products will be uh, wonderful application to the field of agriculture. And the next topic is cell suspension culture establishment. So cell suspension cultures are more advanced cultures than the Callus cultures and offers a wonderful products for the benefit of the human society. Then advantages of the cell suspension cultures, and then conclusion and future prospects of the Callus culture and suspension culture. First of all, what is Callus culture? So plant tissue culture is an in vitro process, in vitro technique that will be carried out within glass or within laboratory conditions. That means aseptic conditions. Every time in vitro means you have to maintain the and a variety of conditions, aseptic conditions where the plant cell can survive and sustain, establish itself, and offers a variety of products, uh, scalable products from the callus tissue. And most probably in vitro technology, in vitro process, in case of plant origin, any plant cells or tissues, organs on a nutrient rich medium, and the nutrient medium contains essential nutrients including macro and micro elements, plant hormones and other components which promotes the growth and development of the concerned plant material on the defined media. See here these are the multiple shoots obtained on a solidified agar medium where micro and macro elements were solubilized and supplemented with the maybe these multiple shoots are obtained because of the increased concentration of the cytokinins and less concentration of the auxins that supports these multiple shoots. Of course, here the multiple shoots can be excised singly and can be re-inoculated on the same media supplemented with the increased concentration of cytokinins and less concentration of the auxins, and again gives rise to complete plantlet. Say meaning complete plantlet meaning in vitro conditions in case of plant origin, it can further develops into organogenesis if you can able to define the cultural conditions, um, a variety of cytokinins like kinetin, zeatin, fiatin, phytin, and other synthetic auxins, for example, 2-isopentenyl adenine, benzylaminopurine, adenine. So these are the synthetic cytokinins, which may every time allows the plant cells, all the totipotential cells especially, and also meristematic cells can be undergone a variety of mitotic divisions and establish the callus, and further the callus can be can establish the multiple shoots, and further organogenesis will be obtained. So how the callus can be uh, initiated, inducted on a defined media? Usually it comprises three phases. Callus initiation takes place, callus proliferation, callus maintenance. Let us see what is initiation, how callus can be initiated on a defined media. Generally, plant tissues which are excised and placed on a culture medium, uh, usually Murashki and Skug medium, any plant origin, either it is a plant cell or a tissue or even organs can be placed on the defined medium supplemented with the designed growth hormones, that is auxins to cytokinins, and sometimes gibberellins also played an important role in sustainability of the culture material on the MS medium. Next, proliferation. After placing a plant tissue or an explant, generally explant is defined as the any excised portion from the mother plant constitutes an explant. So these explants undergo within 15 to 30 days of time under these conditions, generally 25 degrees centigrade plus or minus 2 degrees centigrade, and a relative humidity 70 to 85% relative humidity. And if it is a callus culture, callus proliferation, usually 16-hour dark period and 8-hour light photo period will be applied, and temperature is 25 degrees centigrade plus or minus 2 degrees centigrade, and the pH in the plant tissue culture medium should be maintained, because here callus culture establishment initiation um, only through the maintenance of the cultural conditions, especially the buffering capacity of the medium and also pH of the medium, or played a significant role in designing in sustaining the plant culture material on the MS medium. Coming to proliferation, till now whatever the plant plant origin explant is placed on the under defined MS medium supplemented with the auxin to cytokinins, till now if it is a callus aggregate, prior callus aggregate, so that is completely undifferentiated cells. So during proliferation phase, that is the second phase, the undifferentiated cells start dividing and forming callus tissue. So once they acclimatize to the conditions, here a lag and log stationary, always the undifferentiated cells on the stationary phase, not they may not undergo decline phase. Once it is any nutrient essential nutrients, maybe a major or minor elements missed out, then they may undergo the decline phase, so that the culture the callus culture will be ceased further. So undifferentiated cells start dividing, especially mitotic divisions, and indefinite manner undergoes and forms an unorganized group of cells. In this unorganized group of cells, each and every cell can give rise to a complete plantlet, until unless you design the medium and then formulating the conditions, cultural conditions, then it goes on to develop into organoids and then complete plantlet. Then third phase, that is in the callus culture, maintenance. So every time here the pH and ionic strength is a very important factors, and temperature also. Temperature fluctuations always creates more problem to the callus establishment culture in the plant tissue culture. So that regular monitoring is required after 30 to 45 days during the proliferation. Callus must be regularly monitored. If any browning occurs, or orange red color occur, or black color occurred, immediately the culture bottles or test tubes must be um, removed from the racks or culture maintenance room. Otherwise the other cultures will be contaminated very soon. Since contamination in case of plant tissue culture is a very difficult task, but can be possible through the variety of chemical sterilants, and as well as a little bit of antibiotics also used to control or avoid the further contamination in the racks. So callus is regularly transferred to fresh media for continued growth. If they are maintained on the same medium, that is MS medium supplemented with the previously you have designed auxin rich and cytokinin less concentration, so all the nutrients are completely absorbed by the existing callus cells or inoculated callus cells. So if you are still maintaining on the previous callus medium, the callus cells start moving to the or further proceeding to the decline phase. So here there is a sign if any browning occurs or a cloudy appearance occurs in the callus culture, it's time to change the callus from the um, uh, the previous culture plate or petri plate or culture bottle to another culture plate or culture bottle, so known as the subculturing process. So in case of callus culture, subculturing provides the uh, a variety of products, a variety of callus products or cell aggregates especially, and these aggregates will be fresh and more multiple shoots can be originated if the callus aggregates are transferred onto cytokine enriched medium, less concentration of auxin. Then establishment and maintenance of callus culture. So there are some factors that influence the establishment and maintenance of the callus culture. The first one is explant selection. The initial step involves choosing an appropriate plant tissue for callus induction. This is very important because the generally a matured and or some tender or apical meristem containing plant parts can be selected as a culture material or an explant. So it should be young and healthy and free from contamination. So that's why agriculturists or plant tissue culture technicians or uh, tissue culture agencies always select the tissue with the an optimum concentration of the meristem. So here the meristem plays a wonderful base or significant role in sustaining the callus cells on the MS medium supplemented with the a variety of combination, that is cytokinin auxin rich and cytokinin less concentration. The next factor is the sterilization. So after preparation of the MS medium, all the salts are solubilized and a combination of plant growth regulators were designed, incorporated through the SEAS filtration or filter sterilization, and then the media has to be, after adding the agar and sucrose as a carbon source, the media has to be sterilized either of the moist heat or a dry heat. Of course, usually 99% media, the plant tissue culture media is sterilized through the moist heat sterilization, in which an autoclave or simply a pressure cooker can be used. So that sterilization is crucial for eliminating any microorganisms. So most of the microorganisms that may hinder the callus formation will be removed through the sterilization, sterilizing the medium. So the medium, whatever medium which is designed for callus cultivation, callus culture, should be free from contamination first of all. So the explant and the culture medium are sterilized with a solution of bleach or ethanol. Usually since it is the explant is a young, healthy and free from contamination, should be free from contamination, and the tissue must not be degenerated, because whenever you use the ethanol or bleach, some of the leaf primordia, if it is a leaf explant, or apical tips containing a variety of meristematic cells may be damaged, so that a dilute chemical sterilants are used, for example sodium hypochlorite, magnesium hypochlorite, or less concentration of the mercury chloride can also be used to make surface sterilization of the explant, and culture medium must be sterilized through the moist heat sterilization, that is a pressure cooker or either a stainless steel autoclave instrument, where 121 degrees centigrade and 15 psi pressure and 15 to 20 minutes holding time must be maintained. The holding temperature is here 121 degrees centigrade. So the boiling point of the water can easily generate the water vapor inside the jacket. So the generated water vapor hits the surfaces inside the stainless steel jacket, that is autoclave or pressure cooker. So any microbial forms that adsorbed on the surfaces of the culture material or glassware, metalware, or media, within the media, even though we use the double distillized grade water, the water must be sterilized when since it is used for the media preparation, media composition, so that the media must be sterilized at 121 degrees centigrade, where the all microbial forms will be destabilized with a variety of sterilizing agent. Here the moist heat, the water vapor generates the pressure, so that pressure under 15 psi pressure for a period of the holding period of the 15 to 20 minutes, their cell walls will be completely disintegrated. So once the cell walls are completely disintegrated, the microbial forms are no more microbial forms in the plant tissue culture medium that you prepared for the callus culture, and it is ready for the inoculation. Okay, and the next factor, culture medium preparation. So a specific medium with all the necessary nutrients and growth regulators, that is auxins to ratio to cytokinins, and sometimes gibberellins and abscisic acids also to be included for a variety of objectives, is prepared for optimal callus growth. Then, so once it is prepared, sterilized properly within that 15 to 20 minutes, so after the holding period is elapsed, uh, the media is taken from the autoclave, cooled to room temperature, immediately taken to the culture room. So where the uh, already the laminar airflow chamber is switched on prior the taking the medium from the autoclave to culture room, so which is already sterilized with the UV radiation. Here the physical radiations will be applied on the surfaces of the laminar airflow chamber, laminar airflow cabinet. So here UV rays can directly affect the thymidine molecules of the DNA of the microbial forms, so that indicates, that reveals that the UV rays can directly percolate through the cell wall, that is peptidoglycan of the microbial forms, if it is a bacterial organisms, or a pectin, soberin, lignin, whatever it is, any fungal organisms, or any heteropolysaccharides incorporated as a cell wall or plasma membrane, will be destabilized by these UV rays, especially UVRA, B, C, or the potent rays which generates alpha and beta particles, which disintegrates the stability of this peptidoglycan and as well as the other cell walls of the microbial forms. So in that way, so once the media prepared and sterilized, taken to laminar airflow chamber where already aseptic conditions are prevailed, so the media is always open to the face of the filter, that is a HEPA filter is there in the laminar airflow chamber, high efficiency particulate air filter, at which the inoculation and culturing, subculturing can be performed within the laminar airflow chamber. And care must be taken while you are handling the tissue culture medium sneezing or talking, frequent talking, or eye pricking, or any other unusual activities, and frequent talking or gossiping in the tissue culture laboratories should be avoided, and SOP must be followed, standard operational procedures. Okay, every time all the culture materials or media after sterilization placed inside the laminar airflow chamber, so the technician or the student must be sterilize their hands with the sterilizing agent, that is 75% ethyl alcohol, and also the cabinets after UV sterilization within the laminar airflow chamber, the cabinet surfaces must be sterilized with the 75% ethyl alcohol, and then dry up with the flow at a 0.02 micrometer diameter sized HEPA filter, and then all the culture plates prior sterilized within the either in autoclave or a hot air oven, for example, that is a dry heat sterilization. All the glassware or metalware can be sterilized in the autoclave or in hot air oven. So they must be opened and faced to filter, seize filter in the laminar airflow chamber. So once the media is cooled to room temperature, immediately pour down onto the culture plates, including any culture bottles, standard culture bottles, or boiling tubes or petri plates can be used for pouring the MS medium supplemented with these plant growth regulators. So once they are poured down, moisture must be avoided, since if it is a lukewarm condition, the media always generates a moisture on the upper surface of the upper lid of the petri plates, so once they are completely cooled to room temperature, then only the petri plates are sealed with, covered with the upper lid and sealed with the paraffin film. So leaks must not be there. Paraffin must be properly placed at the edges of the petri plates to avoid any passes of the contaminated air. Of course, within the laminar airflow chamber, it is negligible to, or it is a 1% contaminated air may pass through the edges of the petri plates. Of course, um, the students always sterilize petri plate to petri plate operation to aseptic operation to aseptic operations. The students must sterilize always with the 75% ethyl alcohol to avoid the contamination, and then incubation. So that inoculation, sorry. The sterilized explant is carefully placed on the surface of the culture medium in a sterile environment. Of course, here at the same time, the culture material also taken to the laminar airflow chamber where the culture material is surface sterilized. Generally, the surface sterilization involves the 2 rounds of the ethyl a chemical sterilant, for example, sodium hypochlorite or magnesium hypochlorite or calcium hypochlorite or sodium chloride, sodium hydroxide, or mercury chloride, and immediately followed by the distilled water sterilization. Here, any chemical sterilants that adsorb on the surfaces of the culture material may be absorbed deep into the sections of the culture material and creates more problem to the Callus culture establishment. So that a double distilled grade water is used to remove any trace of the chemical sterilants that absorbed on the surfaces of the culture materials. And right, then incubation. The culture is incubated in a controlled environment, usually 25 degrees centigrade plus or minus degree centigrade maintained. Usually the culture maintenance room is maintained equipped with an air conditioned rooms, and Prior the placing the culture plates or culture bottles or petri plates or culture um, tubes, culture tubes, we 24 hours prior the culture maintenance room is sterilized with the fumigation. The fumigation involves the sulfuric acid, uh, formaldehyde and potassium permanganate crystals. So in an invisible fumes will comes over from the soon after the placing the formaldehyde on the crystals of the potassium permanganate. Invisible fumes come across the culture maintenance room and sterilizes the any spores that are distributed in within the atmosphere. Culture maintenance atmosphere will be completely removed or degraded. So that's why while you are incubating the culture, a control make sure that the controlled environment or not, right? So checking the temperature always must be 25 degrees centigrade plus or minus, and a relative humidity 75 to 85 relative humidity is maintained, and photo period. So since it is a callus culture, the photo period in comprises 18 hour 16 hour dark period and 8-hour light period is applied. And then maintenance of the callus culture. Usually regular subculturing is conducted for callus cultures, periodically transferred to fresh media to avoid the contamination or browning of the media, reddening of the media, and to maintain further maintenance of the growth and avoid the nutrient depletion. Usually maximum nutrient depletions will occur once Callus aggregates still maintained in the previous plates. And then sterile technique. Maintaining sterility is a very crucial point to prevent contamination, which can affect callus growth and introduce unwanted organisms. Any cross contamination sometimes occurs because any contaminated petri plate, if it is not identified properly, all the petri plates in the culture maintenance room will be completely contaminated, and fungal spores can easily distribute, can easily diffuse, even though they are concealed with the paraffin film, but the fungal spores can readily establish their culture even after cooling to the cool the media to the room temperature. Then the optimal conditions. Callus cultures always require a specific environmental condition, that is light, temperature and humidity for optimal growth. So those including secondary metabolites production, genetic engineering, plant propagation, conservation of rare plants. So in case of secondary metabolites, callus cultures are more useful to produce the valuable compounds like pharmaceuticals and cosmetics. Nowadays, all the pharmaceuticals can be cultured or reared or um, cultivated from the either a suspension culture or a callus culture. So most of the cosmetics are plant origin are being being cultivated through the callus cultures. The next one application, genetic engineering. Transformation and regeneration of genetically modified plants can be obtained through callus culture. Next application, plant propagation. Rapid multiplication of elite genotypes. So proper genotypes can be maintained through the callus cultures. Of course, regular subculturing, if regular subculturing is done, so wonderful genotypes can be maintained and preserved for a longer period of time, and also for the commercial purpose. Conservation of rare plants. So this is very wonderful application of the callus culture, preserving germplasm of endangered plant species, and can be transferred from one area, one geographical area to the other geographical area, or one laboratory to the other laboratory, or where the demand is there of one plant origin. So next one is the cell suspension culture. What is cell suspension culture? So these are employed in various applications such as the production of secondary metabolites, genetic engineering, and plant regeneration. So see the picture here. The cultures can be maintained within the liquid devoid of the agar. All the nutrients are suspended, solubilized, and the required concentration of the auxin to cytokinin concentrations are maintained. And cell suspension cultures offers several advantages over callus culture, including more uniform and controlled environment for cell growth and a higher yield of valuable products. So the most advantageous feature of the cell suspension culture over the callus culture, every time whatever product which is being synthesized within the suspension culture or within the cell aggregates can be withdrawn. If it is a reactor, plant bioreactor, or a fermenter can also be used to enhance the cell suspension cultures or to establish the cell suspension cultures. So cell suspension cultures involves the growth of individual cells or small aggregates in liquid medium. This technique is widely used in plant biotechnology and in various for various purposes. Cell suspension culture establishment. So how the cell suspensions can be established involves cultivating plant cells in a liquid medium, allows a higher cell density and a homogeneous growth when compared to callus culture, where cell density may be obtained, but lower cells can always absorb the nutrients, but the upper cells may depend upon the lower cells for the nutrients. And at the same time, homogeneity is missed in case of the callus cultures. Here all the cell aggregates are disintegrated because of they are being maintained in a fermenter like apparatus or a bioreactor, where a baffle or a sparger always circulizes or mixes the culture medium and as well as the cell aggregates, so that each and every cell aggregate will gets the same concentration of or same amount of quantity of the nutrients, so that the aggregates will be increased more when compared to callus cultures. So the process starts by transferring a small amount of callus already cultured from the either previously liquid culture or callus culture to liquid medium. The medium contains all the nutrients including major and minor elements supplemented with the growth element, organic substances, that is vitamins, plant growth regulators, and the carbon source, that is sucrose, either sucrose or maybe directly glucose can also be used. Of course, a mannitol will be used for the suspension cultures, and required hormone combinations, maybe auxin rich and cytokinin less concentration that is necessary for the cells to proliferate. So the protocol involves the cell suspension culture established involves the callus initiation. Of course, the same thing in case of the aggregated callus cultures. Here also callus should be initiated with the increased concentration of auxin, and subculturing of the callus, and then each cell aggregate subcultured part is influenced to form a cell aggregate on the same medium supplemented with the auxin rich, and then each cells are disintegrated and single cell suspensions are maintained in the liquid culture medium. Cell suspension culture plays a very critical role in various applications, including plant breeding, secondary metabolite production. Especially secondary metabolite production is achieved through only cell suspension cultures than the aggregated callus cultures. and genetic engineering. Whatever the new combinations are required in case of the plant species through plant tissue culture, cell suspension culture is the best choice to get the genetically modified products in plant origin. So then after inoculating the cell aggregates, eat or maintain, the maintenance is somewhat difficult, but it is it is possible. It is possible with the cell suspension cultures. Those requires careful attention to several key factors. The culture medium must be regularly refreshed to provide essential nutrients and oxygen while removing waste products. So that the protocol involves the medium renewal. Medium must be, if it is a continuous stir tanked reactor, the medium is added continuously and the product or cell aggregates will be harvested from the medium. pH control. So one of the important factor that that favors the cell suspension culture further in temperature control and aseptic techniques, including always avoid the contamination. So the technician or any gas, unnecessary gas or foam will be generated because of the lower pH conditions or higher temperatures, humidity, high humidity, foam will be generated, must be cleared, so that activated charcoals can be used to control these contaminants, chemical contaminants. And additionally, maintain optimal pH levels, ensuring consistent temperatures are crucial for cell growth and viability. Aseptic techniques are essential to prevent contamination, ensuring long-term health of the culture. Let us see what are the advantages of cell suspension culture. Usually it offers the homogeneity, high productivity, scalability, controlled environment. So let us see each in detail. Homogeneity. So you may not expect the homogeneous cell aggregates in case of callus cultures. They're maintained in the an agar surface. In case of liquid culture, that is cell suspension cultures, always provide a more homogeneous population of cells compared to callus cultures. So that is very important. Once they are homogeneous in nature, each each aggregate can give rise to a complete either a plantlet or each cell can hold the maximum amount of the a secondary metabolite or a pharmaceuticals or a cosmetics. So that is the relation between the homogeneous and the aggregates of the cell suspension. High productivity. The cells in the suspension culture are constantly dividing. Of course, division is also observed in case of the callus culture on agar, but the lower cells always undergo division but won't get the oxygen, enough oxygen or a carbon dioxide gases exchange. But the topmost layers on the agarized cultures, though they obtain the oxygen concentration, they may not absorb the enough nutrients from the agarized surface. But here the cells in suspension culture constantly dividing undergoes mitotic divisions leading to faster growth rate. Higher yields of desired products will be obtained through the cell suspension culture. That is one of the important advantages with the uh, cell suspension culture in case of plant tissue culture. Scalability. Cell suspension cultures can be easily scaled up and until unless they regularly monitored with a skilled person is required to monitor the scalable product in cell suspension cultures. Of course, in bioreactors for large scale production of valuable compounds like pharmaceuticals, drugs, alkaloids, flavonoids, terpenoids, gums, resins. So these are the secondary metabolites. Some spice codiments also be uh, produced on a scalable rates from the bioreactors of plant origin. And then the other advantage with the cell suspension culture, controlled environment, the controlled environment of bioreactors. So maintaining the pH in at a 5.5 pH and 25 degree plus or minus 2 degrees centigrade, and a relative humidity of 75 percentage of the relative humidity always favors the cell aggregates and promotes a consistent high quality production whatever product you require. So let us conclude the cell suspension culture in particular holds a great promise for large scale production of valuable metabolites and biopharmaceuticals. Of course, further research and development in this area can be performed to get the improved production or process or optimized protocols, innovative protocols, and knowledge sharing from the advanced laboratories, and increased yields and reduced cost can be uh, obtained. So as the field of plant tissue culture, plant cell culture continues to advance, so one can or we can expect to see exciting new applications and innovations in the coming years. Let us summarize the present chapter, that is Callus culture and suspension culture. Callus culture is obtained through the unorganized group of cells on a defined media. Suspension culture can be obtained without an agar on a defined media. So both are essential techniques in plant cell culture, each serving different purposes and offering unique advantages. Callus culture is pivotal for inducing undifferentiated growth and subsequent differentiation, while suspension culture is ideal for large-scale cell proliferation and metabolic production. Together, these methods support various applications in plant research, biotechnology, and commercial production, and especially those products being manufactured, being cultured in callus culture or economic importance, commercial significance. So that the callus culture is a very important for the commercial firms, those who are practicing or commercializing their products on an indefinite manner. Cell suspension culture very particularly holds a great promise for large-scale production of valuable metabolites and biopharmaceuticals. Further, research and development in this area can lead to improved production process, increased yields, and reduced cost. So these lines are very important for the plant tissue culture, especially callus culture and also cell suspension culture. Of course, cell suspension culture is more advantageous than the cell callus culture with respect to the agar, with respect to the conditions, especially hormonal combinations. So in case of the suspension culture, where the secretion, indigenous secretion of the hormones, especially IEA, and less concentration of the cytokinins always enhances the large number of cell aggregates in the suspension culture, so that all the aggregates or each and every cell has a great surface area to get absorb the available nutrients in the cell suspension. As the field of plant cell culture continues to advance, we can expect to see exciting new applications and innovations in the coming years. So these are the references for the present chapter, that is cell culture, that is cell callus culture and cell suspension culture. So go through the references for further studies, further clarification about the chapter. Thank you for your great attention in the present chapter.